Je vais juste commencer comme ça, mais dans un mode un peu express, juste pour donner un peu le ton. Je m'appelle Christophe Villot. Et la première question est, quel est votre métier? OK, Matthew, where's Matthew? Aha. So, what is your job?
Hello, everyone, and thanks for the invite here. Very happy to see you on this snowy day. So my background is a bioinformatician. I guess you could also call me a computational biologist. So my job is very similar to what a data science does, except I'm involved essentially with biology. We're a very disciplinary team, so I work with physicians and neuroscientists, and essentially we're decoding the genome. We're looking specifically, I'm at the Douglas Mental Health Institute, so what we're interested in is maternal health. We're looking at how the perinatal, so pregnant women, and how that period affects early childhood development. We use a lot of information. I mean, in the last 10 years in genetics, we've kind of made leaps and bounds as far as the different types of data we're getting. We started off with, decoding the genome sequencing. We've moved on to getting some more important questions into the functionality of this, looking deeper, like what does DNA methylation do? What does histone modification do? How is gene expression affecting different things? So we're interested in merging a lot of different data sets as well. We're dealing with some unstructured data like photos, so MRI imaging. We're interested in integrating some biometrics through personal sensors like Fitbits in the future. So really my job is to kind of we work in a team about 20 people. We have about 6 computational people, and we're making close connections with the people above us, running a lot of data. And so some of the issues that we have in data is just the large scale of it, trying to integrate these various different data sources together. A lot of my time is spent trying to get unruly data together. You hear a lot about data munging, so I'd say a lot of my job is involved with actually cleaning up the data before I can run anything on it. I mean, a smaller percentage of my time is spent doing statistical analysis or machine learning or visualization, although I think it's a really important issue. That's why we're all here, visualization. I think it's kind of like this 80-20% rule. I spend about 20% of my time on visualization, perhaps. But I think that like 80% of the effectiveness is being able to describe this information to the people that I'm working above me. When you start working with like physicians, like I said, you kind of realize that, okay, I need a way to translate this research, and visualization plays a very important role in this as well. So thank you. Oh, actually, could you just go one slide? I wanted to show you my workplace as well here. So this is just kind of a view of my home. I don't have this many consoles at work, although that's my Christmas present that I'm asking the boss for. If any of you work in R, it's really cool trick if you don't take anything else away from today. You can detach the console, so you can kind of make your life easier and have, I like a lot of consoles, console heaven. So yeah, this is basically, I wanted to just mention like what I use for it. I use typically all my work is like in the terminal. A lot of the bioinformatics tools that I leverage are all in there. That's why I use this. A lot of the data manipulation and visualization that I do is within R. I've dabbled a little bit in Amazon cloud computing in the past. However, now I'm working with and I'll get into this later, but now I'm dealing with humans. I used to be more in the animal science. And with genotyping data, this is personal data we don't want to get out. You've heard of like some of these leaks and from 23andMe and these kind of public companies. So all of the work we do is on institutional clusters. And I think that's all I have to say. Thanks, Chris.
Excellent. You can stay here if you want. Thanks a lot. Matthew, please, for a project, Fitbit project.
I was actually going to do the...
Oh, yeah, right.
So anyone can read this later. I was talking earlier about integrating some biometric data. This is already up on my blog. I threw together this slideshow over the last week here. I think this would be a little bit more interesting for you guys. Feel free to check out my blog later. Okay, so as I mentioned before, communication is really important, whether this is going to talk to an executive, your boss, or anyone else, it's really important to have good visualizations. Recently, so I'm going to highlight some of the projects that I've been involved in. Recently, I was trying to come up with a great way of showing gene expression changes in the brain. So we're interested in looking at depression in pregnant women. And if we have gene expression data and we know it's linked to a certain brain region, it's interesting to be able to show this with 3D brain visualization. So what we have here is I came up with 3 different methods for generating brain regions. One of them is BrainNet, Mango. These are Java and MATLAB implementations. Essentially, this is a composite image from T1 MRI images. They take it from about 30 different people, so you get an average perspective of what is a typical brain. Then we use what's called a brain atlas. These have volumes for about 90 different regions in the brain that we know of currently. And so basically, I was able to overlay these brain atlases with a mask, and I'm able to highlight certain regions of interest. So for here, we have the cerebellum, or here we have the left hippocampus. And I thought this is really nice. They're very visually appealing. It's a great thing that we can include in publications. The problem with these, they're GUIs, so they're graphical user interfaces. And this is kind of hard for reproducing stuff. I actually ran into the issue where I saw this in a paper that came out in Nature, and I was really interested in replicating it myself. When I went to approach the authors, they didn't have good documentation on how to do this. And when I approached them, they essentially said RTFM, read the effing manual, which was a bit difficult for me. So it took me like 2 weeks to figure out how to do this on myself, and I ended up blogging about it so future researchers can do it as well. But the problem with graphical user interfaces is, you have to take so many screenshots to show someone how you would do this. I wanted to do this in R programmatically so that it would be a little bit more easier to reproduce. The code for this I'll eventually have up, so anyone interested in looking at it. This is a brain image. Now the skull has not been stripped, so you can still see that the head is around here. We can get a nice visualization from different aspects of the brain. If we look over here, we kind of get a freaky vision here as we can see the eye sockets. So I mean, this is a kind of a neat way. This is probably like 20 lines of code for doing this, and we can highlight different regions of the brain and change the coloring of this based on how strong that expression of gene signatures is. Similar to this, I was also playing around with this GG anatogram thing, and much like the 3D brain images, we're able to label different systems. So we've got nervous system, respiratory system, reproductive systems, and we can color these however we want to kind of highlight and bring attention to certain areas. And you can do this for different model species. So in my lab, I primarily deal with humans, but we also have part of the lab that's dealing with model organisms like rat and mice. So together, I was going to use this package and this other one called GG Genes, which is showing essentially gene arrows. I'm sorry, it's a little bit blurry here, but it shows, the start and the end, whether these are in different genomes or on different chromosomes. And I wanted to leverage this to create an image that we were going to be submitting for a grant in the near future. So here I've got 2 different females, and you can see that the brain region has been highlighted differently in this area. So let me give you a little bit of background on this. Your genetic sequence is ACTG, 4 letters that make up your code, and you have the same genome from birth to death, except if you get like a mutation, but it's pretty much stable. Now, epigenetics, if you've heard the word before, is a layering on top of the DNA that can be changed throughout life. So this is really interesting for treating cancer or other different diseases or phenotypes. And so we know from studies where there's been childhood abuse or traumatic events like post-traumatic stress disorder, where we can tell from the methylation pattern, so this is epigenetic effects on the DNA, that persist throughout lifetime causing problems. And we know that this methylation causes aging in the body, so that we can use this kind of information to get what's called like an epigenetic age. So for example, if you're an alcoholic and you're drinking all the time, your liver is going to take a hard toll. So your liver, the cells are going to look actually older. You might have like a 40-year-old liver if you're 30 and drinking hard for a long time. In cases where we don't have methylation, because this data is very difficult to get, we can make inferences. So what we're trying to do is use a proxy of looking at 116 estrogen hormones in the genome, and we want to look for these areas where transcription factors bind. This is what proteins that are recruited to the machinery that cause other genes to be put out. So we see here in this instance that we have a difference between the A and the C over here. So this is what we call a SNP, a single nucleotide polymorphism, and this leads to different phenotypes, so in this case, depression in females. And we want to use this as a proxy. So I mean, this is a great way of making an infographic with a couple packages that we can send on to grant reviewers to get them. So what are these CPG islands? CPG islands are essentially regions of the genome where there's a lot of C's and G's close together. Now, only the C's are methylated, and when these are methylated, it stops the transcription factors from binding, and these proteins are not expressed. We know in cancer that when these genes have methylation, that tumor suppressor genes, genes that are meant to stop cancer, are no longer active, and this leads to cancer. So in our bodies, we have about 28,000 islands scattered throughout the genome. This is a representation with the karyoplotter showing an ideogram geome here of chromosome 9 and the distribution of different CPGs around. Now, we typically like to do these different plots where we kind of show different genetic regions. This is leveraging the complex heat map package, and it adds a little bit more depth to it. So it's kind of domain specific in the fact that we're looking here near transcription start site. This red line up here shows where the transcription start site is in the trough, and then regions upstream and downstream of that start site. And we can see regions on the y-axis. These are different samples. So for example, in areas where people might have more cancer. This has a little bit more functionality than what you would see in your typical heat map. You can do complex annotation, and it uses also some R statistical stuff, so clustering based on like k-means, for example. Another kind of popular plot I want to show you that's really big and might have some relationship, you might have seen this in your own domains, is called the Circos plot. So the idea was it was really difficult to show these kind of translational effects and similarities between genomic regions linearly. So what they came up with at the Michael Smith Genome Center in Vancouver was that you can put these in a circular manner. So here we have on the top the entire human genome, chromosomes 1, 22, and the X and Y on the top, and then looking at dog chromosome 31. And you can find areas that are similar in dog chromosome 31 match up with chromosome 13 and 4 in humans. Now this is just a very crude one that I did by hand because I actually haven't got into Circos myself, but I am interested in getting into it. This is a more complex diagram that was produced not by myself, but by 23andMe. As you can see, it's very complex. This is why I wanted to show it. You can layer a lot of different information. We've got links going on here. We've got histograms. We've got heat maps. Variety of stuff that you can put on here. So just diving a little bit closer into this, showing different things. As I mentioned before, there's the 22 chromosomes going around as well as the X and Y. They range from about 0 to 50,000 base pairs, and some of them, like this, or up to 250,000 base pairs. And this is showing different similarities between these regions and people that have different SNPs throughout their body, or areas, for example, here, where it's showing these locations are associated with different disease, like diabetes, or mental, or any kind of phenotype. You can have Alzheimer's, or there's cancer up here. These links are showing similar areas that have similar biochemical pathways throughout it. And then in the colored sections, we see that these sections are areas in the genome that have very similar sequences to other areas. So this is kind of a neat way of, it's a high ink to data ratio, so you're able to put a lot out there in one single graph. When you're limited and only, say, putting 4 figures into a manuscript, you're able to convey a lot of information. Packages to create this. This was invented, like I said, from a lab in Canada. So they've got a graphical user interface. I'm not a huge fan of that. You can use a couple different R packages to do this. These are static images, though. You can also use JavaScript for an interactive web-based visualization of that. I want to go into a few more interactive things. I know Nicholas is going to be talking about Plotly later, but this is called the Manhattanly package, and it's used for GWAS, which is called Genome-Wide Association Study. So essentially, we're looking at all of the SNPs that are different between control and a disease phenotype. This is built on top of the QQMan package and the D3JS engine. So it's just going to take a few seconds to do, but I've got an interactive plot here. It's called a Manhattan plot. Now, typically, there would be more. You would see tons of these things. It's kind of reminiscent of the skyline of Manhattan, and where we see these large skyscrapers from the buildings that are a little bit lower and varying around the city. All of these are SNPs between individuals. On the x-axis, we have all of the chromosomes and the base pair positions between this. And on the y-axis is the negative log p-value. So essentially, the ones higher up are more statistically significant, saying that you have a higher function in that disease or phenotype in these. So the nice thing is when there's lots of these points here, you can mouse over to see which SNP is associated with which gene. It makes it a little bit easier when you're sharing it with other people to look through your data. As well, now we've got an idea of, okay, these are the genes that have effect. We want to look at how important are these genes. So this is what's called a volcano plot. I don't know if any of you are familiar with the Bland-Altman plots. It's also called Tukey mean difference. It's to look for differences between, say, if you have different tools measuring something, or you want to look at retest reliability for a certain instrument, you use this. And this has been co-opted for genetics especially. So on the bottom, we have like a regression coefficient, log fold change, odds ratio. Similarly, it's the log 10 value, log p10 value. So anything that's higher, and in these regions here, will be stuff that has a large effect that we may be interested in. And I mean, this is a nice thing about JavaScript and these web-based implementations, is that I can send this off to a senior researcher. They can have a look at it at their own time, come back to me when they're ready, and we can talk about the results of the test personally. Plus, I mean, a lot of these things, I'm not from a neuroscience background, so these weird names and numbers, I'm not familiar with all of them. Biologists, they love to look at these lists, so it's great for them. Helps them look for what they're interested in the data. So if there's any questions, you can ask me later, I suppose. And this was created with Sharagan. I was kind of interested in finding a way where I could use R to implement all this stuff into a PDF slideshow. You have a question?
Yes, I was wondering whether any utility is still using ABS, or is it considered loading software?
Which one?
ABS Express.
I haven't used ABS Express. No.
So it must be devoted.
I'll have to look into that. It might have been
Google it and tell.
AVS.
It was the standard.
Okay.
I suppose used less and less now.
Okay, I have not heard of it. Yes?
It's not a question, but that was a very good presentation.
Thank you very much. Appreciate it.
So, what was your career path?
Thank you. So I had a little bit of a roundabout way getting into programming. I actually started off trained as a marine biologist and ecologist in statistics. And I kind of made my way into programming through that. I've always been interested in design, and I remember we used to have these wet labs where we were dissecting fish, and a partner of mine, she would do these really great illustrations. Well, I can't draw to save my life. So when I go to talk to people and I'm trying to explain a concept, don't get me in front of a whiteboard because I can't do it. So visualization has always been really cool to me. One of the first projects I worked on was in MATLAB, and I was looking at taking videos and turning these into batch set of images that we could look at. We were looking at sponge sneeze responses. They sneeze much like humans to get rid of it. That kind of spearheaded into a project. I worked with Fisheries and Oceans, did some consulting, freelance for a while, and we were looking at remotely operated vehicles taking pictures under the water for BC Hydro. They installed a cable, and we wanted to see how did this affect these sponge reefs, which are thousands of years old, and they can't sneeze like freshwater sponges. They're made out of glass spicules, so they die as soon as they're covered with sediment. So I came up with some image-based analysis looking at regions of interest. We would stitch together these large panoramic images of hundreds of images together over a transect along the water and look for, okay, what was the baseline before they installed this to the future, and how did that affect the health of the community, of this reef community. That really got me interested in programming. You know, I was, before that, I was collecting a lot of the data myself, but I was really interested in analyzing the data and being able to show people about this. So then I moved into genetics. I heard, you know, this is kind of, there's a good field out there for bioinformatics. If you've ever heard, there's not much out there for marine biologists. That's very true. But I really found a passion in it. And so I did some wet lab work for a bit, realized that I really didn't like being behind the bench and doing that. I really like being behind the computer, analyzing data. So the last little while, I spent a year at the Austrian Academy of Sciences in Vienna, and there I was more involved in oncology and diabetes research, dealing with some proteomics, transcriptomics, and now I've kind of moved into the neuroscience department, so it's always fun learning about new things. We've got these new visualizations, like I've showed you with the Manhattan Leap project for GWAS studies, which I'd never done before, so I'm always, you know, interested in learning about new things. I guess, where is my career going now? You know, Nicholas was talking about making things easy for people without a JavaScript background. I'm not a JavaScript person, but I was in collaboration with some people who've designed a really nice way of visualizing categorical data. And what I'd like to do with them over the coming months is a collaborative project to essentially use the R2D3 to leverage these JavaScript visualizations, bring it to the R community, which is quite big in science, and, you know, kind of marrying these two fields together. So that's kind of where I'm heading now.
What are the advices that you would give?
Okay, I'll make this really quick. Just kind of respond to that point. I guess it's about a community. I think if you're going to be practicing, you might as well put yourself out there, blog about it. Not only does this help solidify it in your own mind, you're going to help other people learn stuff along. I think it's great to be part of these communities. I think that you should kind of make yourself a deliberate like curriculum for things that you want to achieve and kind of mark these off the list. If there's weaknesses that you don't have and you want to add these to your skills, try and focus on that. Maybe some visualizations that you haven't done before. Don't be afraid to ask for help. Like what I think is a lot of people with a growth mindset, they're not worried about how they're going to come across. They're not like afraid to say, oh, I don't know the answer to this. I like to have like a 15-minute rule at work that if you don't figure something out, ask a colleague for help. Don't be afraid of like them looking down at you that you don't know this. You're going to save your boss's time, your own time, your colleagues' time. Reframe that to be like, I'm missing a learning opportunity. So whether this is, you know, responding to stuff or asking questions on Stack Overflow or whatnot. And then, you know, when you get further along, share your knowledge, right? Like I said, like learning this stuff yourself, you're not only solidifying it, but by doing this, you're going to help other people learn as well. And this helps everyone out along.